Hello, and welcome to the Chapter 18, Part 3 lecture on Eukaryotic Gene Expression. You should use the information in this lecture to complete the Chapter 18, Part 3 guided notes, which, of course, you should complete before you attend class. In Part 2 of this chapter, we discussed how prokaryotic cells such as E. coli can use operons and regulatory molecules to turn their genes on and off. In this part of the chapter, we're going to discuss how eukaryotic cells turn their genes on and off. Now remember that both the genetics and the gene expression pathway in eukaryotic cells is far more complicated than we found in prokaryotic cells. There are more steps involved in the eukaryotic pathway. So that means that there are many more opportunities for turning these pathways on and off, and that the means of controlling gene expression in eukaryotic cells are going to be much more complicated. Eukaryotic cells can activate or deactivate the gene expression pathway at many points. They can affect how genes are expressed at the level of transcription by affecting how that gene is copied. They can also affect the transcript, that little mRNA copy that's made. It can be modified in different ways. And finally, they can affect any of the steps after transcription, which we call post-transcriptional control. There's also something called the epigenome and the genetic hierarchy that can play a role in turning genes on or off. One of the things that makes eukaryotic gene expression so much more complicated than prokaryotic gene expression has to do with the fact that eukaryotic cells are not very organized. In part 2 of the chapter, we learned that prokaryotic cells have pretty organized DNA. Their DNA is fairly short. It's organized into one little circular chromosome, and the genes are organized into operons. Operons group genes with related functions together, and it puts a single promoter in front of all of those genes. So all of the tools for a certain job are grouped together. Now in eukaryotic cells, it's different. Eukaryotic cells have many chromosomes, many long stretches of DNA. For instance, inside a human cell, there are 46 chromosomes. And the genes with related functions aren't grouped together. They're spread all over the place. And each of those genes has its own promoter. So each individual gene has its own switch to turn it on and off. That means that you're going to have a much more complex system of regulatory molecules to turn these genes on and off, especially when you're trying to do a job that requires several genes. The major control element used in the prokaryotic system were these stretches of DNA called operators. And if you remember, the operator was the portion of the promoter to which repressors and activators could bind to turn those genes on or off. Now, eukaryotic cells use promoters as well, but they have a single promoter in front of each gene, so every gene gets its own promoter. And in addition, they have many other types of control elements, other little regions to which a team of regulatory molecules are going to need to bind in order to turn transcription on or off. This makes the system much more complex. Most of the molecules that bind to the control elements in eukaryotic systems are referred to as transcription factors. Transcription factors play a big role in forming the transcription initiation complex, which, if you remember, is the machine that allows transcription to proceed. Here we can see this in action. We have all these little control elements, we have the promoter, and we have the gene itself. Here you can see various transcription factors beginning to bind to different pieces of this DNA and form this machine, this complex. This is the thing that RNA polymerase number 2 is going to bind to, and once everything is in place, it can begin transcribing that gene. If all of the transcription factors are present and are able to bind properly, that turns the gene on. However, if any of these transcription factors are missing or are misshapen for various reasons, that's going to turn the gene off. So eukaryotic cells control whether their genes are on or off largely by controlling these transcription factors. Enhancers are one of the weirdest control elements used by eukaryotic cells, so we definitely want to take a look at them. Enhancers are weird because of their location. Look at where it is. It's way over here, whereas the gene and the promoter are way over here on the other end. We say that enhancers are very distal, which means distant in science. Now, in order for the enhancers to do their job, these little activator proteins have to bind to them, and then somehow they have to reach the promoter region over here and lock into place with the other elements. To accomplish this, this other weird little protein has to bind to the DNA, and it actually bends the DNA. It curls it over so that the enhancers can come in from the top and lock into this transcription initiation complex machine.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Different types of cells are different types of cells because they make different transcription factors and different regulatory molecules. Consider this. Both liver cells and cells in the front of your eye have identical DNA. They both carry a gene for a protein called albumin and another called crystallin. Albumin is important in liver cells. So liver cells make the transcription factors and have the regulatory molecules necessary to turn the albumin gene on. However, the crystallin gene, which makes the clear front of the eye, is not important at all in liver cells, and in fact would probably be a problem if it were expressed. So liver cells don't make the transcription factors to turn that gene on. The opposite is true inside lens cells, the cells on the front of your eye. These cells don't need albumin, so they don't produce the regulatory molecules or transcription factors to turn that gene on. So it's off most of the time. But they definitely need crystallin to build that clear front of the eye. So they do produce transcription factors that will allow them to transcribe the crystallin gene and make that crystallin protein. In a previous chapter, we discussed how mutations or changes in the DNA within a gene can affect a specific protein product. By changing the DNA sequence, you can change the shape of that protein and thus alter its function. Now consider this. Control elements, such as promoters and enhancers, are also just sections of DNA. and so they can pick up mutations as well. When you change the switch in front of a gene, you can potentially alter when and how it makes protein, and that can really cause some problems for the cell. For instance, here we have a little promoter that's picked up a mutation, and because of this mutation, the gene has turned on when it's not supposed to be. And because of that, that gene is making way too much of this protein product. Many different types of cancers are caused by this exact type of phenomenon, where genes are turned on that are not supposed to be turned on. Eukaryotic cells continue to make decisions about the gene expression process even after transcription has already occurred. We call this post-transcriptional control. Let's say that the gene has already been activated, transcription has already occurred, and this little mRNA transcript has already been formed. Now, what should the cell do with it? Well, that depends on the needs of the cell. It can decide to send it on to translation, and if it does so, it needs to decide specifically which proteins it wants to make from that transcript. Or it can decide to stop the process of translation, and there's a couple of different ways it can do that. Before translation can occur, the cell needs to decide specifically which protein it wants to make from that transcript. It can do that using that process that we discussed in the last chapter, called alternative RNA splicing. In alternative RNA splicing, the cell decides which little sections of the transcript are going to stay in and which sections are going to be cut out. For instance, over here, sections 1, 2, 3, and 5 have been kept in. We call the sections that are kept in exons. And section number 4 has been cut out. We call that an intron. In this little section, exons 1, 2, 4, and 5 have been kept, and section 3 was cut out as an intron. So by deciding which little pieces to snip out, it can alter the protein product that it ends up with. In addition to containing genes that code for proteins and control elements that regulatory molecules can bind to, DNA also contains sections that code for these things called non-coding RNAs, or ncRNA molecules. Now, we've talked about RNAs that are used in the protein making process. So these RNAs go through transcription, they are transcribed, and then they're used in translation to make proteins. But these guys, the non-coding RNAs, only go through transcription. They don't go through translation. So these RNAs don't carry the code for making an amino acid sequence. Instead, they fold up on themselves, and they have a number of really strange little shapes. They may be spliced or changed by the cell, but they do not go into translation and are not used to make a protein. Non-coding RNAs have been recently discovered. They have a number of different functions inside cells, but one of the processes that they participate in a lot is the control of gene expression. So these non-coding RNAs help turn different genes on and off and help control the products of transcription. So let's say that a cell has been busy all morning making a particular protein. It's made a bunch of transcripts, a bunch of Xerox copies of that particular gene, and it sent them out, and they're being translated over and over and making lots of this protein. But then the cell decides to change its mind. It doesn't need that protein after all. What can it do to reverse this process with all those little transcripts floating around out there? Well, that's one of the roles that some of these non-coding RNAs can play. There are RNAs known as microRNAs, or also RNAs known as small interfering RNAs, or siRNAs. And what the cell can do is it can start producing these RNAs in large amounts. These RNAs are going to be complementary to the little transcript that was already made that the cell no longer wants. And so these little complementary strips will float around, they will find the little piece of RNA that's no longer wanted, and it binds to it. And it only binds to the RNA that it's complementary to. So that's how it can be kind of specific in which ones it's going to target. Now, once these RNAs have bound to the transcripts, those transcripts are either going to be broken down and recycled, or sometimes they just don't enter translation. So they kind of block up the process so that the ribosome can't grab onto that transcript. This silences the process. It reverses the process by making the transcripts unusable. Let's say that a cell has been busily making protein all morning, and then it decides, I don't really need this protein anymore. Well, what's it to do? It's already finished translation, and these proteins are already floating around doing whatever they do. How can you get rid of them? Well, at that point, we call that level of control post-translational, because it's after translation has been complete. And there are a couple of really neat little molecular machines that can help out with this. One of them is a signaling molecule called ubiquitin. Ubiquitin can be attached to the little protein that we want to get rid of. And then the attachment of that ubiquitin allows the protein to be taken in by this big crazy machine. This crazy machine is called a proteasome. It's shaped a little bit like a big tube, and you can see that the protein with its little signaling molecule fits inside. And then this machine, the proteasome, acts like a little molecular wood chipper. You feed something in there, you turn it on, and it breaks down that protein and spits out all the little amino acid subunits. It spits out all the chips. It's kind of fun to imagine it making a really loud, fun grinding noise. But this allows the cell to recycle protein even after it's gone all the way through the process of translation, and it prevents proteins from doing an unnecessary job if they need to be deactivated. Another means of controlling gene expression has to do with how tightly or loosely the DNA is wound. We refer to this area outside the DNA as the epigenome, and this means of control is called epigenetic control. If you remember, when the cell isn't dividing, the DNA is going to be stored in a fiber called chromatin. In chromatin, the long strip of DNA, each chromosome, is wound around these little proteins called histones. And collectively, a histone protein plus a little section of DNA that's wound around it is called a nucleosome. Now, if these nucleosomes are wound loosely, that means that enzymes can get in and work with the DNA and express the DNA, the genes in that region. However, if the DNA is wound very, very tightly around the histones, it means that enzymes can't get in there. There's no room for them to fit between those nucleosomes and work with the DNA. So when the DNA is tightly wound, the genes in that area are turned off. When the DNA is loosely wound around the histones, gene expression of those regions is turned on. So regulatory molecules can be used to bind to control elements, and this action can turn genes on or off. In addition to that, though, we have this whole giant system of molecules that work specifically on the epigenome, and their job is either to tighten the DNA in a certain region or loosen it, and that can also help turn genes on or off. It's a second level of control. When certain molecules, such as acetyl groups, are bound to the DNA, it has a tendency to loosen the DNA in that area. It loosens up the nucleosome. Actions such as deacetylation can tighten them back up. Methyl groups can also loosen and tighten DNA. When methyls get together with phosphates and bind to a piece of DNA, it tends to loosen the DNA, and those genes tend to be expressed. But if methyls are joining to the DNA by themselves, it tends to tighten the DNA, and so those areas of genes are silenced or turned off. Eukaryotic genes seem pretty disorganized. Genes of related functions aren't located next to each other in the eukaryotic system the way they are in the prokaryotic system. Instead, genes of related functions are spread out all over the chromosomes, and each individual gene has its own promoter, its own on-off switch. However, just because these genes are spread out from one another doesn't mean there's no organizational system at all. In eukaryotic cells, we refer to their organizational system as the genetic hierarchy, and it actually works a little bit like an employee hierarchy in a business. On the top of the hierarchy, we have the president of the company. Okay, these genes that we call master genes. And what they will do is they will send out instructions, they will send out molecules that will activate other genes. These genes in the middle of the hierarchy are kind of like managers. Those managers are then going to tell the workers what to do. They're going to say, hey, start expressing your product, and those products need to get to work. Now, the signals that the master genes on the top tier send out tend to be fairly generic. For instance, let's say it's a construction company, and the big boss has said, let's build a house. All right, well, there are a lot of little jobs that need to be done in order to build a house. The big boss needs to activate the managers need to organize the actual workers. And then these individual workers are going to do jobs like work on plumbing, electrical work, framing, maybe some of them put the roof on, all of the little jobs that you need to do to build a house. Another great example of master genes are these genes we call Hox genes, also known as homeotic genes. Homeotic genes have been studied extensively in fruit flies. So fruit flies are going to be our example here. Now, homeotic genes are active in all animals when they are embryos, so only during embryonic development. And what these genes do is they send out messages that help segment the embryo into different parts. In other words, there are genes that are active on the front that tell that part it's going to be the head. There are genes that are active in the middle, in the thorax area, if we're talking about a fruit fly, and that tells it it's going to be the thorax. And there are genes that are active in the end of the fruit fly that tell it it's going to be the abdomen. So the Hox genes determine which part of the embryo is going to be which part. Now remember that the Hox genes are master genes. They are the boss genes, and as such, the messages they send out are very generic. The Hox gene that tells the thorax of a fruit fly to be a thorax just sends out the message, build a thorax. And then that activates genes in the middle that are managers, and they are going to organize their specific workers to build the thorax. The thorax of a fruit fly contains things like legs and wings and some internal organs that are kind of important. So all of these structures have to be grown in the right places in order for this fruit fly to be normal. Now the Hox gene at the top, it really has no idea how to grow a leg. That's not its job. It just sends out the signal, build a thorax. It's actually the little worker genes at the bottom tier that know how to build fly legs and put them in the right place. One of the very first Hox genes to be characterized is a gene known as the Eyeless Gene. Now, the name eyeless is a little bit deceptive. The scientists who discovered the eyeless gene called it eyeless because when they turned the gene off in fruit flies were born that didn't have any eyes. So they called it the eyeless gene. Really, this gene is for eyes. When this gene is expressed during embryonic development in a fruit fly, it will grow an eye wherever that gene is expressed. So that's a normal fruit fly with a normal eye over there on the left. Now let's take this a step further. What if we activated this gene in other parts of the body? Could we grow eyes in other parts of the body? Well, that's exactly what the scientists did. Here you have a fruit fly that has normal eyes. So the eyeless gene was present and working when it was an embryo. But look right here. There's a little tiny eye growing on the base of the wing. What the scientists did was activate this Hox gene in the cells at the base of the wing, and so a tiny little eye grew in that spot. Now, the fly can't see out of that eye. It's too tiny, and it's on the wing. It would be flapping around all the time. But it's kind of an interesting proof of concept. Many other mad sciencey sounding experiments have been done with Drosophila fruit flies and these Hox genes. Here's another good example. On the left here we have a normal fruit fly with normal antennae growing out of its head. On the right, the gene that expresses legs has been turned on where the antennae normally are. So you get these big crazy long legs growing out of the head. All animals have Hox genes, and interestingly, it turns out that the genes used to build a fruit fly's body are actually really similar to those used to build a mouse's body, and those genes are really similar to those used to build a human's body. Now, Hox genes were discovered long before gene sequencing was a thing. So how in the world did they figure this out? Well, there's an interesting experiment. What the scientists did was design a mouse embryo in which the eyeless gene was turned off. So this little mouse, if it was allowed to grow to term, would not have eyes. Then the scientists took the Hox gene for eyes from fruit flies and injected it into that little embryo. Now think about the possibilities. It might be that the fruit fly gene just doesn't work in the mouse system, and then you'd have a mouse born with no eyes. Or you might have a mouse born with fruit fly eyes. That would be exciting. Or you could have a mouse that's born with normal eyes. The mouse that was born had normal eyes. Now, what does that tell us? Well, that tells us that the fruit fly gene must be really similar to the mouse gene, or else the cells wouldn't recognize it. It also tells us that that Hox gene sends out a really similar message in fruit flies as it does in the mouse embryo. This only works because that Hox gene is a master gene. So the message it sends out just says, build an eye here. It doesn't specify what kind of eye. It doesn't tell the body whether to build a compound eye or a regular light seeing eye, as we find in mice. So what happened was that the little Hox gene activated, and then the genes that work for that Hox gene got busy, and they said, oh, okay, build an eye. We know what kind of eye to build. We're going to build a normal mouse eye. The discovery of Hox genes has some pretty interesting evolutionary implications as well. Think about a classic example of evolution. Let's think about a fish that over many generations develops limbs for walking on land. That's a lot of physical change. Now, scientists knew that that physical change results from changes in the DNA, so that there are many different little mutations that have to happen in order for fins to eventually change into limbs. However, they didn't think that the mutations could happen fast enough to cause the kind of physical change they saw in the fossil record. The math just wasn't adding up. However, the discovery of Hox genes changed all that. Because Hox genes are in charge of many other little worker genes, a mutation in a Hox gene can cause a lot of physical change all at once. In other words, if you make a change in a Hox gene that has to do with building fins, it might tell its workers, hey, we're not building fins anymore, we're building limbs now. And so they get to work and they build limbs instead. So the discovery of Hox genes really helped tie the entire evolutionary theory together. It made it make more sense. An interesting example of a Hox gene in the human system is the Sry gene. Now, the Sry gene isn't a homeotic gene. It doesn't have to do with segmentation of the body, but it still has some pretty powerful effects. Female embryos in the human system have the chromosomes X and X, while males have an X and a Y chromosome. Now that little Y chromosome carries this Sry gene. And this Sry gene at a certain point in development is going to send out the message, make the body male. So all human embryos essentially start out female. Female is kind of the default setting for human beings. And then if that Sry gene is present, it activates at a certain point and it turns the genitalia into male genitalia. If that little gene is not present and is not activated, then you end up with the female system. All right, you guys, you're finished with chapter 18. Isn't that an interesting chapter? I really like that one. Make sure you use this information to complete your guided notes and bring them to class, as well as any questions that you have. We're going to work with this information some more.